Hi, good evening everyone. I hope you all are doing well. Welcome to the third marathon class on Botany Insider. Let's wait for the students to join in and then we'll start off with our class, okay? Good evening, Sambhavi. Good to see students coming back to our marathon classes every day. Sambhavi, you are regularly joining all the sessions. That's excellent. Join in on our app as well. Let's wait for just 2 more minutes, then we'll start off with our lecture, okay? Students are joining in, let them join, then we'll start. Okay. Is the audio and video clear to everyone? Just let me know, please. Just confirm audio, video, whether the app is like the presentation is visible to you. I am audible clearly. Just confirm. Good evening to all the students joining in. So today is the third day in our lecture series, and today we are going to discuss about one of the most important concept of CSIR. If you look into the syllabus, that is the Operon model. We'll try to cover up both of them, like both the LAC Operon and the TRYP Operon. Even if by anyhow, if we are done with the time and we are left with the topic, do not worry, we'll complete it or we'll carry forward the topic to the tomorrow's class, okay? So do not worry about it. And these topics are important for all the exams like be it CSIR, DBT, ICAR, GATE, any exam which you are targeting. All these topics, like every topic is very common in the examinations, right? So Operon model is one which is like which can be studied just once and it would be covered for all the classes. Okay, now let's start off with the class. It's already 8:03. So welcome all the students who have joined. So before starting, if you haven't yet downloaded our application, please download it from the iOS and from the Play Store both. It is available on both. In addition to that, we have a lot of content available there. We have our notes booklet, we have the courses running. New course starts on 11th of December. So if you wish to enroll in that, you can enroll in that as well. It would be a crash course for the students who are targeting the upcoming exam. Okay, so in addition, we have the general aptitude course which is ongoing. The PYQ pointers are available. We have the notes booklet as well. Good evening, Harshit. Good evening, Farheen. Okay, so now let's discuss about the concept of Operon. Okay, so today we'll first understand like what Operon is all about, because ये exam के अंदर बहुत recurring topic है, so it's good to get this concept cleared. Okay, now let's talk about like what an Operon is all about, because we have been discussing about कि Operon model को study करना है. But what is Operon? Because पहले इसकी knowledge is very important. Now what is an Operon all about? So it is a set of functionally related genes in the bacterial cells that are frequently clustered together in a single transcription unit. Simple meaning, let's not go into the such a hi-fi language. Very simply, what is it? It is a set of genes, like you can see in the figure. These are the genes. So these genes are clustered together in a particular format. For what? Because why are the genes actually used for? Genes are actually used for getting the final product, because obviously in a transcription, because if you have seen or if you have read about the Operon model, or if you have read about basically the central dogma of biology, it says the DNA gets converted into RNA by the help of what? By the help of transcription, and RNA gets converted into the proteins by the help of translation, right? So DNA is carrying what? It is actually carrying the genes. This is what is written over here. That is, operon is what? It is a set of functionally related genes that we are talking about, the DNA only. DNA would undergo transcription, as in the case of our central dogma. This is what the central dogma actually defines us. So with transcription, it would then be converted, or it would then be translated into what? Into the protein, which is our final main work that we need to do, right? So operon is what? It is the same thing. We just have a set of the genes that are arranged in a specific manner, and they would be transcribed and then translated to form the proteins. So depending upon which genes are there, we have majorly two type of operons. We study about the LAC operon and we study about the TRYP operon, and these operons are in the bacterial cells. Okay, now there were two main French biologists that have actually proposed this complete work. That is, they have given this particular concept of the operon for the regulation of the gene action in the E. coli, who were they? Francis Jacob and Monod. Okay, so this was about it. Now let's understand about the various terminologies or the various terms that are being used in this complete operon concept, because time and again you'll see a lot of terms getting used or repeated again and again. Let's just understand about it. The first one is operator. So what is an operator? Or before that, look into this particular image, you will get to understand. You will see a promoter being here. You will see a promoter, you will see operator, regulatory genes, structural genes. All of these terms are over here. So we need to understand the meaning of the same. The first one is what? The first one is operator. Okay, so the first one is operator. What is an operator? So operator is a DNA sequence in the operon of the bacterial cell to which a regulator protein binds, and this binding affects the rate of transcription. Let's understand it in simple manner. See, there is a complete sequence of events going on in which you have the DNA. DNA gets converted into RNA and RNA into protein, right? So one thing that you have to understand is, for this DNA to start off with the transcription, what is actually required, or what is the main purpose of it? It need to start off, like we know that okay, the transcription need to be there, but from where should it start? From where, or onto which particular point should it start? So this is what the operator is. So what is an operator? It is the main area, it is the area where our first main component, our first regulator protein would come and bind to, and after its binding, what would happen? This binding, because when the regulator protein comes and binds to our promoter, as in this figure, just look at here, you have let's say this is a regulatory protein, it would come and bind to this operator site. So when it comes and binds to this operator site, then the main rate or the main speed by which this complete process has to happen would be decided. Okay, so this is what the operator is. Second is what? Second is our promoter. So what is a promoter? Because see, what is the word itself give the meaning. Operator is what? Something that is operating or something that is initiating the work. What is a promoter? Promoter would be the one that would be actually making the work get to start in a specific manner. So DNA sequence in the operon of the bacterial cell to which the RNA polymerase binds and initiates the transcription. Then we have the inducer. What is an inducer all about? So an inducer, it is a substance that stimulates the transcription in an inducible system of the genes. That is usually a small molecule that binds to the repressor protein and it alters the repressor as well. See, The word itself says inducer, or we can say induction. So what do we mean by the word induction? Something that leads to the initiation of something. So inducer is the one that would be stimulating the transcription. Then we have the regulator gene. So what is a regulator gene? So regulator gene is the one that is associated in an operon that encodes a protein or a RNA molecule which functions in controlling the transcription of one or more structural genes. So this is what a regulator is all about. So I hope all these four main terms are clear to everyone. If you have any doubt, any issue, you can ask me. Otherwise, we'll move forward. Any doubt? Not the promoter of one gene binds to the operator of other. We have a promoter, we have an operator. These are both the part, or these are both the main structures in our structural gene. In this complete operon, you have a promoter and an operator present. So we'll be having a different protein, a different regulatory protein, which would be coming and binding over here and starting off the main work and starting off the main thing. Okay, so I hope, Harshit, your doubt is clear. Let's move forward now. Now let's talk about the types of operons. So we majorly have two types of operons. First one is the inducible one, second one is the repressor one. So as the word itself suggests, operator, inducible and repressor. So what does these mean? Inducible, so that means the one that, or the operator, that operator system that normally remains switched off condition and begins to work as and when the substance is there to be metabolized. Then we have the second one as the repressor. So what is a repressor? The one which is actually normally in the on condition, but it has to be switched off as and when the work need to be stopped. So inducible means the operator normally in on condition, normally in the off condition, and it starts to work when the work is actually, or when the substance that is there which is need to be metabolized. For example, the LAC operon. Then we have the repressor one. Repressor is the one in which the operator molecule, which is normally in the on condition, but it would continue to produce its substance, it would continue to produce its metabolite till the time, or till the amount when it is present, or like till the time it is required by the cell. But as and when the requirement gets done, it turns into the off condition. So inducible, initially off, starts to work as and when the work needs to be initiated. Repressor, initially in the on condition, but as and when the work is done, it needs to be stopped off. For example, the TRYP operon. This particular concept is very, very important about inducible and repressor. If you have any doubt, please ask. Any doubt till now? Because this particular point is very important about the type, like about the inducible and the repressor one. If you have any doubt, please ask. Any doubt, students? If no, then we'll start with the first one, that is our LAC Operon. Okay, so let's start off with our very first one, that is the LAC Operon. So why is it called as LAC Operon? It is from the term lactose. Okay, how and why are we actually studying this? Let's understand. See, when we look into the nutrition of the bacteria, so this bacteria have the preferred source as glucose. That means, let's understand it simply. Let's consider this is the bacteria that we have. Okay, now this bacteria could have two sources of nutrition. First is glucose, second one is lactose. It is similar to the case like you must be like on a table, let's say that you have pizza and burger in front of you, but you must be having any of the two, from the two any one of being more favorite, right? So when we look into the same thing for the bacteria, so for bacteria the glucose is a more favored, or we can say the glucose is more favorite to the bacteria. That is, if in any media we have glucose and lactose, both of them being present, the bacteria would first prefer to have the glucose consumed for its nutrition. But as and when the glucose gets done, then उसके पास कोई ऑप्शन नहीं होता ना कि अब अगर मेरा एक source of nutrition is over, now what is required or what can the bacteria actually do? उसको even if it is not a preferred source, उसको lactose uptake करना पड़ेगा, right? So this is the main thing that you have to understand. Just have an example of pizza and burger in front of you. Let's say you have burger and pizza. So for you, let's say the pizza is more favorite, and if I ask you which one would you prefer more, so you would for sure say pizza, right? But as and when the pizza is complete, what would you do? You would not be having any kind of option in front of you. You would have to have burger for yourself. So same, apply the same thing to the glucose and lactose. If bacteria have in the same media glucose and lactose, both of them being present, glucose is the pizza for the bacteria. It would first have glucose eaten, consumed, and then it would start to consume the lactose for its nutrition purpose. Okay, this is what is written over here. See, we have the preferred source being the glucose, that if glucose is present, the bacteria will first utilize it. But if glucose is absent, as the case I have just told you, that if the glucose is absent and lactose is present, what will happen? Lactose will be metabolized into glucose and galactose. We know that lactose is what? It is a disaccharide. And which are the two subunits of lactose? The first one is glucose and the second one is galactose, right? So as and when the glucose gets consumed, as and when the glucose gets completely consumed in the nutrition media, then the lactose would be consumed and it would be metabolized in the same manner. It would be broken down into glucose and galactose. Okay, so this is the complete LAC Operon concept is based upon this preferred source only. If you understand this, then the remaining portion becomes very, very easy. Now see, if the E. coli breakdown of lactose, when available, it requires 3 enzymes. That is, in E. coli, if the lactose is present, what will happen? It would have to be metabolized, it would have to be broken down. So for breaking down that thing, what is required? We require 3 main genes. Which are those? We have gene Z, we have gene Y, and we have gene A. That means Z, Y and A. These are the 3 main genes available. What are these? Z, Y and A. So in the E. coli, let's say the thing that we have just understood, let's say in the nutrition media, E. coli was there, glucose and lactose both were present. Glucose is first understood, utilized, now we have lactose present. So for metabolizing, for using these lactose, or we can say for metabolizing this lactose into glucose and galactose, what is required? We require three main genes. Which are those three genes? It is gene Z, gene Y and gene A. So which are the three structural genes for our LAC operon? We have three main genes as gene Z, Y and A. Remember about this. Now this E. coli and the other bacteria, they metabolize the glucose preferentially even in the presence of lactose. This is something that we have just understood. But what is the reason for that? Because it requires less energy. Obviously, glucose is the simplest form of sugar, right? For galactose, what is required? Sorry, for lactose, what is required? We require more amount of energy for that, right? So why would bacteria do more work in the initial go when it know that it have an easy source, it have glucose in front of it, it can easily use glucose. So when it's having glucose, it would use it because it requires less energy for its modification. So remember about this as well. So this is about the main thing. Then addition or the presence of the lactose itself stimulates the production of these required enzymes. All these genes are actually the enzymes. They would be coding for a specific enzyme. Which one? We'll just understand. Wait for it. Then we have that lactose is a negatively inducible operon. What does this mean? We'll just understand. So see, very, very important point from here. Glucose, it is the primary source for the energy metabolism. We have just understood. When the glucose is scarce, scarce means when it is present in less amount. So the bacteria induces the synthesis of the protein for what? For actually utilizing the lactose, because when glucose is present in less condition, the lactose have to start off with what? It would have to actually start off with the utilization of lactose, and this lactose would now work as a energy source. The lactose has three lactose utilization genes, which are these LAC Z, LAC Y and LAC A. Now over here it's very, very important to understand or remember actually what are these. Just a second. Yes, we have LAC Z, LAC Y and LAC A. Lactose Z encodes for what? It encodes for beta galactosidase, which breaks down the glucose. Beta galactosidase also convert lactose into allolactose. So remember about this point. Then we have LAC Y. So it encodes for what? It encodes for lactose permease, which is doing what? Which allows the lactose to enter the cell. And finally we have LAC A. So LAC A is the enzyme that would encode for the trans-acetylase, which helps in the inactivation of what? Of inactivation of thiogalactosides by acylating them. Please remember about each of the term given to you. Very, very important. Every single thing written on my slide, written over here, is important. All the classes that we are conducting, all of these classes will help you. All you have to do is make notes sincerely and be very, very attentive in the class. Okay, then adjacent to the structural genes there is a common promoter present, and when the LAC operon would be turned on, when it would be turned on as and when the lactose is present and the glucose is absent. Okay, so this was about it. If you have any doubt till here in this particular slide, you can ask me. Otherwise we'll start off with our next slide. Any doubt till now? Photosynthesis portion have been already covered on YouTube in a very full-fledged manner. If you like go through my complete YouTube channel, you will see there are lot of videos for photosynthesis in a playlist as well, and also in the revision format. So please watch that. It will be easy for you. Okay, Harshit, what about others? Please let me know. Okay, Madhu. Let's move forward now. If there's no doubt, we can move forward. Let's do one thing. We have done it in the live also. You can watch the recording for the live, and if you want to study in a structured manner, we have a course as well. LAC Z, okay. See Pinku, for the LAC Z, what is it? देखो LAC Z के अंदर it encodes for what? It encodes for beta galactosidase. What is the main requirement for the, or what is the first requirement for lactose? Just let me know. For lactose, what do we require? We need to break it down, right, into glucose and galactose. So LAC Z would be helping in this. It would be encoding for the beta galactosidase, and this would help in what? It would help in the breakdown or the metabolizing of the lactose into the glucose and galactose. Is it clear, Pinku, now? I hope it's clear now. Now let's move ahead and understand about other things. So see, when the glucose levels are high inside, the lactose utilization enzymes are not needed. Very, very easy. See, that means when the glucose level are high, because obviously we have a glucose and a galactose present, right? So if it is glucose present, so the galactose utilizing enzyme won't be there for sure. So in this particular case, what is required? We need to shut it down. We need to shut down the operon by the LAC repressor, which binds to the operator which is present adjacent to promoter. Because see, why are we actually having this complete promoter and operator present? We need to initiate any work, but when the glucose is present, lactose requirement or lactose genes are not required. So a repressor would come and it would bind over this particular position, which would lead to the inactivation of the complete process, right? So this is what is written over here. Now next we have the LAC repressor block the binding of the RNA polymerase. Now if you have idea about the fundamental processes in which we study the complete transcription and translation, so for transcription we require the binding of RNA polymerase. So this lactose repressor would bind to, it would bind to the operator region where it would be blocking the RNA polymerase activity, and as and when the RNA polymerase activity gets blocked, that means transcription would not happen and the further processes would be inhibited. Now as and when the glucose level falls, because there is a particular limit of glucose present or a particular quantity present, as and when the glucose starts to fall down, the levels, the galactose is detected in the medium. Because if you have a platter in front of you and when you have a pizza and a burger both, and your favorite is pizza, you won't be noticing burger till the time you get finished with pizza, right? So this is the same thing, right, happening over here as well. So as and when the pizza for the bacteria gets over, what would happen? It would start to think of, let's have the burger as well. So the burger for the bacteria is what? It is the lactose. So now the LAC operon would be turned on and it produces the enzyme that are required to break the lactose present, right? Now a small amount of beta galactosidase is constitutively present that converts the lactose into allolactose. Now see, is it possible for a bacteria to like start the work at just a click? No, right? So for that, what does a bacteria actually does? It keeps on producing some amount of beta galactosidase in its cell, like very, very small amount, so that the LAC operon, if it wish to be turned on after a point of time, it can be turned on instantly. So for that, a small amount of beta galactosidase is always present. Then what it would be doing? It would be converting lactose into allolactose. Now what is a allolactose? For the LAC operon, allolactose behave as an inducer, and if it is behaving as an inducer, and we have just discussed the main function of inducer, what it would be doing, it leads to the initiation or the transcribing of the genes that would require or that are required for the metabolic process of our lactose. Okay, so allolactose will be working as inducer. So this is how it actually works. Look at here, you have the in the absence of allolactose or inducer. So what is the inducer for lactose? It is allolactose. Allolactose. So if the allolactose is absent, what would happen? You have the inducer, promoter, operator and the LAC operon, 3 genes, right? When it is absent, what would happen? The repressor would keep on binding to the operator and it would lead to the blocking of our LAC operon. But if the inducer is present, the inducer would come, it would remove the repressor and the complete work or the complete transcription, or we can say the main process of the operon would initiate. That is, the Z would be producing beta galactosidase, Y would be causing the production of permease, and A would be causing the trans acetylase to be produced in the bacterial cell. Now then we have the two main things over here. Just a second. Yeah, now we have the catabolic repression of the lactose. So what is catabolic repression all about? See, catabolic repression is that when or in what way do the operator have to actually work. When the glucose is available, the genes that participate in the lactose metabolism are turned off through catabolic repression. Simple meaning that when the work is not required or when the process is not required, the lactose operon should be closed, and for that thing there is a specific term given to it, that is catabolic repression. Till here, if you have any doubt, any problem, you can ask me. Otherwise, we can first discuss the doubts for your. Is there any doubt? Yes, any doubt till now? Other students, is there any doubt? Okay, very good. Let's move forward then. Now, see, this catabolic repression, it results from the positive control response to glucose. What does this mean? This means that because of, or how does a catabolic repression actually occurs? When glucose is present, it would lead to the positive catabolic repression, because what is it? It is the blockage, or we can say it is the stoppage of the functioning of LAC operon, right? So if the bacteria is positively regulating to glucose, so what we would say that the catabolic repression is positively regulated, or it is in the positive control in response to the glucose. Now the positive control, it is accompanied by the binding of a activator protein called as CAP, which is catabolite activator protein. What does this mean? Glucose is present, right? You have the glucose present. So for this, what we are saying that it is a positive catabolic repression. How does it occur? It occurs by the binding of a protein given the name of CAP, that is catabolite repressor or catabolite activator protein. As and when the CAP is present, or when the CAP protein is present, RNA polymerase cannot bind, and because RNA polymerase cannot bind, activation cannot occur. Because CAP also act as a glucose sensor. It activates only when the glucose level is low. That means CAP will also act as a glucose sensor, because this would get activated as and when the glucose levels are low. It would be removed and the work would be completed. So CAP senses the glucose indirectly through the hunger signal, which is the CAMP molecule. Let me explain this to you in simple manner. What are the components we have studied? We have studied about catabolic repression, we have studied about CAP and CAMP and glucose, right? See, when there is high amount of glucose present, what will happen? There would be positive catabolic repression. Catabolic repression occurs because of the presence of the enzyme because of the presence of the protein CAP, catabolite activator protein. Now this CAP protein, it is a indirect sensor of glucose. That means when the glucose level gets low, it would get removed by the help of hunger signal, and this hunger signal is CAMP. Because see, जब बैक्टीरिया के पास ग्लूकोज की कमी होगी तभी हमें लैक्टोज को एक्टिवेट करना पड़ेगा. लैक्टोज के एक्टिवेशन के लिए CAMP की रिक्वायरमेंट है. कैप प्रोटीन ग्लूकोज के कम लेवल को एक्टिवेट करेगा बाय द एक्टिवेशन ऑफ दिस. इनडायरेक्टली कैप जो है वो बैक्टीरिया को हेल्प कर रहा है टू स्टार्ट ऑफ विद द लैक्टोज मेटाबॉलिज्म. व्हाई? बिकॉज कैप प्रोटीन ने सेंस कर लिया कि ग्लूकोज कम है, वो जाकर CAMP को एक्टिवेट करेगा. CAMP आकर कैप को हटाएगा, कैटाबॉलिक सप्रेशन नेगेटिव होगा, ग्लूकोज का लेवल कम हुआ, लैक्टोज का लेवल बढ़ा और ZYA जीन्स एक्टिवेट होकर लैक्टोज को ब्रेक डाउन करेंगे ग्लूकोज और गैलेक्टोज में. हंगर सिग्नल डिग्रेड हो जाएगा फिर से, बिकॉज नाउ द बैक्टीरिया हैव फूड टू eat, or we can say bacteria have lactose to get metabolic energy and start off its further work. Clear everyone? This is bit tricky, I know, but I have tried to explain you in simple manner. If there's any doubt you can ask me. Just go through this once again. It's very simple. Just try to understand the main concept behind it. Read this and listen to the content explained to you. It would become easy. Okay? Clear everyone? Yes, any doubt? Great. Very good. If there's no doubt, that's excellent. So what I am thinking of, that let's stop the class over here, because from here there is more technicalities starting and I just don't want you to get burdened by this complete information. So let's do one thing. We'll start off with from this particular slide in our next marathon class, okay, so that you can just go through every single thing till here, and in addition to that you can watch this slide, then this particular slide, and then we have two mutation slides. You can just go through it from your end as well. In the next class we'll discuss the remaining portion and then we'll start off with the TRYP Operon, because if we'll go through all these things, then it would become a lot of like a burden of information for you all. Okay, So let's do one thing, let's end the class over here, let's just keep the section till here. You can make your notes till here, and then we'll start off with this slide in our next marathon class. Okay, should we do it this way? It would be easier for you. Just try to analyze what you have learned, make your own notes, and use the further slides as well. Like I have already shown you these slides, you can complete out the topics with yourself, and any doubt or anything, because whatever the main thing was we have discussed, there are two things left which we'll be discussing tomorrow or in the next marathon class. Okay, till here let's keep it, and in the next class we'll meet. Now it would become easier for you to get through this complete concept. It's a bit difficult. We'll complete the other topics tomorrow in the next marathon class. Okay, so was the class helpful for you people? Yes. And are you people liking this marathon series from Botany Insider End? Other students, are these series helping you out? Not just CSIR, many other exams could be targeted with these questions and with these topics, because in every single exam the pattern of exam might be different, but most of the time the main important thing is that you have to go through the conceptual understanding. Okay, so we have a lot of other contents available on our application. Please download our application. You can enroll in our courses. New course is going to start on 11th of December. You can enroll in that. It is a crash course for you all, so make sure that you join in there. We also have a general aptitude series running on the application. Join in there, it would be helpful for you. So thank you so much everyone for joining in. Let's meet in the next marathon class. Okay, take care everyone, and do not forget to like, share and subscribe to Botany Insider. Bye everyone, take care.